So we are continuing with part 2 of immunodeficiency, where now we will be talking about secondary or acquired immunodeficiency. We'll have something like 1 hour and also with this discussion, and it will be a nice discussion and intensive one. Yeah, and mostly we'll talk also about the HIV. You need glucose for nice reading experience. In the water. So we continue. Secondary acquired immunodeficiency. So we want to discuss on secondary or acquired immunodeficiency. And here our discussion will deal mostly to acquired, and HIV will be the main topic. So deficiency of immune system often develop because of abnormalities that are not genetic but acquired during life. Table 2 to table 21.6, which is just there, it says there are secondary acquired immunodeficiency causes and mechanism. Like when you're speaking of HIV infection, the main cause is the depletion of CD4 T cells. In protein calorie malnutrition, metabolic dearrangement inhibit lymphocytes maturation and function. On irradiation and chemotherapy cancer, decreased bone marrow lymphocyte precursors also can explain the process. On cancer metastasis and leukemia involving bone marrow, the mechanism is reduced site of leukocyte development. On immunosuppressive transplant or autoimmune diseases, the mechanism is reduced lymphocyte activation, cytokine blockage, impaired leukocyte trafficking. On the disease of loss of spleen as a result of trauma, sickle cell disease, or surgery, here there is decreased phagocyticity of blood-borne bacteria. So those are main things that you will be discussing actually, that we will be talking in this chapter. So secondary acquired immunodeficiency. Deficiency of immune system often develop because of abnormality that are not genetic but acquired during life. Acquired immunodeficiency disease are in fact more common than congenital immunodeficiency and are caused by a variety of pathogenic mechanisms. First, immunosuppression may occur as biologic complication of another disease process. Second, so-called iatrogenic immunodeficiency may develop as complication of therapy for other diseases. Third, immunodeficiency may result from infection that target cells of the immune system. The most prominent for these is HIV infection, which is described separately later in this chapter. Disorder in which immunodeficiency is frequently complicating elements including malnutrition, cancer, and infection. Protein calories malnutrition is common in low-income countries and associated with impaired cellular and humoral immunity to microorganisms. Much of the morbidity and mortality that afflict malnutrition people is due to infection. The basis of the immunodeficiency is not well defined, but it is reasonable to assume that a global metabolic disturbance is the disturbance in this individual caused by deficient intake of proteins, fat, vitamin, mineral will adversely affect maturation and function of the cells of the immune system. Patients with advanced widespread cancer often are susceptible to infection because of impaired cell-mediated and humoral immune responses to a variety of microbes. Bone marrow tumor, including the cancer metastasis to bone marrow, and leukemia that arise in marrow may interfere with the growth and development of the normal lymphocyte and other lymphocytes. In addition, tumor may produce a substance that interfere with lymphocyte development or function. Various types of infection lead to immunosuppressions. Viruses other than HIV are known to impair immune response. Examples include measles virus and the HTLV, human T cells lymphotrophic viruses. Both viruses can infect lymphocyte, which may be basis for their immunosuppressive effect, like HIV, HTLV1, retrovirus with a tropism for CD4 T cells. However, instead of killing helper T cells, HTLV1 transform them and produce aggressive malignant neoplasm called adult T cell leukemia or lymphoma, ATL. Patients with ATL typically have severe immunosuppressions with multiple opportunistic infection. Chronic infection with the Mycobacterium tuberculosis and virus fungal frequently result in anage due to many antigens. Chronic parasitic infection may also lead to the immunosuppression. For example, African children with chronic malaria infections have been depressed T cell function. This may be due to one reason why these children have increased the propensity to develop Epstein-Barr virus associated malignant tumor. Iatrogenic immunosuppression is most often due to the drug therapies that kill and functionally inactivate lymphocytes or block the function of cytokine made by innate immune cells and lymphocytes. Some drugs are given intentionally to immunosuppress the patients, either for the treatment of anti-inflammatory disease or to prevent rejection of organ allograft. The most commonly used anti-inflammatory and immunosuppressive drugs are corticosteroids and calcineurin inhibitor, respectively. But many other, including anti-cytokines antibodies, are widely used now. We have already seen it in chapter 17 and chapter 19. Various chemotherapeutic drugs are administered to patients with cancer, and these drugs are usually cytotoxic to proliferating cells, including mature and developing lymphocytes as well as other lymphocyte precursor. Thus, cancer chemotherapy is almost always accompanied by a period of immunosuppression and increased risk for infection. Iatrogenic immunosuppressions and tumor involving the bone marrow are the most common cause of immunodeficiency in high-income countries. Another form of acquired immunodeficiency results from the absence of spleen caused by surgical removal of organ after autoimmune hemolytic anemia and thrombocytopenia, in which red cells and platelets, respectively, are destroyed by phagocytic in the spleen, or infection in sickle cell disease. Patients without spleen are more susceptible to infection by some organisms, particularly bacteria such as pneumococcal and meningococcal, which have polysaccharide-rich capsules and are more cleared by opsonization and phagocytosis. This enhanced susceptibility is partly due to defective phagocytic clearance of the microbes in the blood, an important function of the spleen, and partly due to the defective antibody response resulting from absence of marginal cell B cells. Human immunodeficiency and acquired immunodeficiency syndrome. AIDS is a disease caused by infection with HIV and is characterized by a profound immunosuppression with associated opportunistic infections and malignant tumor, wasting, and neurocognitive disorders. HIV primarily infects activated CD4 helper T cells. Macrophage and dendritic cell can also harbor the virus but do not do so less efficiently than activated CD4 T cell. HIV emerged as a human pathogen recently relative to the most other known human pathogen, and the HIV endemic epidemic was first identified only in 1980s. However, the degree of morbidity and mortality caused by HIV and the global impact of this infection on health care resources and economics are already enormous and continue to grow. HIV has infected 70 to 80 million people and has caused the death of more than 34 million adults and children. Approximately 38 million people are living with HIV infection and AIDS, of which approximately 70% are in Africa and 10% in Asia, and almost a million people die of the disease every year. An especially devastating feature of this disease is that half of the approximately 3 million new cases every year occur in young adults, 15 up to 24 years of age. AIDS has left approximately 14 million orphans, about 1.7 million children under 15 have HIV, and more than 90 of these children acquired AIDS from the mother during fetal life, childhood, or breastfeeding. About 100,000 children die of AIDS every year. Currently, there is no vaccine or permanent cure for AIDS, but antiretroviral drugs are in use and that are very effective at controlling the infection. In this section of the chapter, we'll describe the properties of HIV, the pathogenesis of HIV-induced immunodeficiency, and the clinical and epidemiological features of HIV-related diseases. An overview of HIV virology. HIV is a member of lentivirus family of animal retroviruses. Lentiviruses including Visna viruses of sheep and the bovine, feline, and salmon immunodeficiency viruses are capable of long-term latent infections of the cell and short-term cytopathic effects, and they all produce slowly progress fatal disease that include wasting syndrome and CNS abnormalities. Two closely types of HIV designated HIV-1 and HIV-2 have been identified. HIV-1 is by far more common cause of AIDS. HIV-2, which differ in genomic structure and antigenicity, causes a form of AIDS with slower progression than HIV-1 linked disease and is mainly confirmed in West Africa. HIV structure and genes. An infectious HIV particle consists of two identical strands of RNA packed with a core of viral proteins and surrounded by a phospholipid bilayer envelope derived from the host cell's membrane, but including viral encoded membrane proteins. The RNA genome of HIV is approximately 9.2 kilobase long and has a basic arrangement of nucleic acid sequences characteristics of all known retroviruses. You see it in figure 21.4. Long terminal repeat LTL at each end of genome regulate viral genome expression, viral integration into the host genome, and viral replications. The GAG sequence encodes core structure proteins. The ENV sequence encodes the HIV envelopes, ENV proteins. ENV exists as a trimer of 3 molecules of glycoproteins, the glycoprotein 120 and a stem of 3 molecules of glycoprotein 40 that is inserted in a viral lipid bilayer. The Pol sequence encodes reverse transcriptase, integrase, and viral protease enzymes, which are required for viral replications. In addition to these typical retrovirus genes, the HIV-1 genomes contain 6 other regulatory genes unique to HIV, namely as TAT, REV, VIF, NEF, VPR, and VPU genes, whose product regulate viral replication and host immune evasion in various ways. The function of these genes are summarized in figure 21.4 and discussed later. Viral life cycle. HIV infection of the cell begin when the envelope glycoprotein GR120 of the virus binds the 2 protein on host cells. The CD4 are usually a chemokines receptor that function as co-receptor. The viral particle that initiate infections are usually in the blood, semen, or other body fluids of one individual and are introduced into another individual by sexual contact, needle stick, or transplacental passage. The 2 components of ENV, the transmembrane GR41 subunit and the external non-covalently associated GP120 subunit, are produced by proteolytic cleavage of a GP160 precursor. The trimetric ENV complex mediate a multi-step process of fusion and a fusion of the virions envelope with the membrane of the target cell, is seen in figure 21.6. The first step of this process is binding of GP120 subunit to CD4 molecule, which induce the conformational change that promote secondary GP120 binding to a chemokine receptor, generally CCR5 or CXR4, which function as the co-receptor for the viruses. HIV binding to the co-receptor induces a conformational change in G41. Is refolded in 6 helix band that exposes a hydrophobic regions called a fusion peptide, which then insert into the membrane and enables the viral membrane to fuse with target cell's membrane. Fusion may occur at the cell surface or in macropinocytic or endosomal compartment. After the virus completes its life cycle in the infected cell, described later, free viral particles are released from the infected cells and bind to an infected cell, thus propagating the infection. In addition, the GP120 and GP41, which are expressed on the plasma membrane of infected cells before virus is released, can mediate cell-cell fusion with an infected cell that expresses the CD4 and co-receptors, and HIV genomes can then be passed between the fused cell directly. For the ones who will follow, you can follow this lecture again in the, I'll put the video that is showing the picture of what I'm showing now. So actually, the HIV figure 21.3 structure of HIV-1, An HIV virion is shown next to T cell surface. HIV consists of two identical strands of RNA, the viral genomes, and associated enzymes including reverse transcriptase, integrase, and protease, packed in the cone-shaped core composed of P24 capsid proteins with a surrounding P17 protein matrix, all surrounded by phospholipid membrane envelopes derived from the host cell. Viral encoded membrane proteins GP41 and GP120 are bound to envelopes, CD4 and chemokine receptor on host cell surface function as HIV-1 receptor. And this here, we follow, this is the human host cell, and these are CD4 receptors actually, and these are T lymphocyte surface, and these are chemokine receptor. You remember the receptor, we said there is CCR5 and CXR4. So here we have P17 matrix, which is inside here. We have RNA, and inside we have a reverse transcriptase. We have protease, and also we have integrase. We have P24 capsid. Then outside there is a bilipid layer, of which most of the time we are told it is from the host cell. So they are these ones like anchoring things, GP120 and GP41, this one. So that is the structure. So HIV genome, the gene along linear genome are indicated as differently colored blocks. Some of genes use some of the same sequence as other genes, as shown by overlapping blocks, but are read differently by host cells RNA polymerases. The coding sequence at the TAT and RAV genes are separated in the genome and required RNA splicing to produce functional messenger RNA envelope GAG group specific antigens. So those are things that someone only who can see the screen then can follow. So what is happening, the figure 21.5 is HIV life cycle. The sequential steps in the life cycle of the HIV are shown, and from initial infection of the host cell to viral replication and the release of the new virion. For the sake of clarity, the production and release of only one new virion are shown. And infected cells actually produce many virion cells, each capable of infecting cells, thereby amplifying the infection cycle. Proviral transcription is activated by cytokines or antigen, not shown. So in here, it seems that we have HIV virions, then virion binding to CD4 and receptor. After they have binded to the chemokine plasma membrane and chemokine receptor, the fusion of HIV membrane with cell membrane entry of viral genome into cytoplasm happen. The viral genome after entering is HIV RNA genome, then with the help of reverse transcriptase, the synthesis of proviral DNA. Then it will happen in the integration of provirus into the cell genome. In the nuclear, then they will kind of integrate HIV DNA provirus and HIV transcript. Then from there, they will be produced HIV proteins. This synthesis of HIV proteins assembly of virion core structure. Then HIV core structure, expression of G120, G41 on cell surface, budding of mature virions. This is again going outside the cell. So here, cytokine activation of the transcription of HIV genome transports the spliced and unspliced RNA to cytoplasm. Then it moves out. The most important chemokines receptor that act as co-receptor for HIV are CCR5 and CXCR4. More than 7 different chemokines receptor have been shown to serve as receptor for HIV entry into cells, and several other proteins belonging to 7 transmembrane spiny G proteins coupled receptor families such as these leukotriene B4 receptor can also mediate HIV infection of the cell. Different isolates of HIV and indeed different HIV strain within the same individual have distinct tropisms for different cell populations. Although CD4 T cells are the main cellular target of HIV, some strain of HIV that have IMF protein that possess a very high affinity for HIV can infect macrophage as well. These strains are sometimes called macrophage M tropic strain. The density of CD4 on macrophage is 20 times lower than it is on CD4. The density of CD4 on macrophage is 20 times lower than it is on CD4 T cells. And the expression on the macrophage cell surface of CCR and CXR4 is also considerably lower than level of these proteins on CD4 T cells. Although genetically distinct viruses exist in an infected individual, typically only a single viral strain is successfully transmitted from an infected individual to previously uninfected one. The transmitted viruses is called the founder strain. The founder strain that are transmitted from one person to another have relatively low affinity for CD4 and exclusively use the CCR molecule as co-receptor, the CCR5 molecule as co-receptor. The strain called T-trophic strains enter the macrophage about 30 times less efficiently than M-trophic strain. Person-to-person transmission almost always involve only T-trophic strain. The importance of CCR in HIV infection in vivo is supported by the finding that individuals who do not express this receptor on the cell surface because of inherited homozygous deletion in the CCR genes are resistant to HIV infection. After an HIV virions enter the cells, the enzyme within nucleoproteins complex become active and begin viral reproductive cycle. You have already seen in figure 21.5. The nucleoprotein core of virus become disrupted and RNA genome of HIV is reverse transcribed into the double-stranded DNA of viral reversible transcriptors and viral DNA into the nucleus. The viral integrases also enter the nucleus and catalyze the integration of viral DNA into the host genome. The integrated HIV DNA is called provirus. The provirus may remain transcriptional inactive for months or a year with little or no production of new viral proteins or virions. And this is a way HIV infection of an individual cells can be latent. The contribution of latent viruses to HIV reservoir is discussed later. So we have the diagram for the one who will see. We have the diagram which is the mechanism of HIV entry to the cell. In the model depicted sequential conformational change in GP120 and GP41 are induced by binding to CD4. These changes promote binding of the viruses to co-receptor, a chemokines receptor, and fusion of the HIV-1 and host cell membrane. The fusion peptide of activated G41 contain hydrophobic amino acid residue that mediate insertions into the host cell plasma membrane. So we have virions, there's CD4 here, host membrane, the chemokine receptor, then HIV membrane, G41, GP120, CD4 cell membrane, HIV GP binding to the T cell and the CD4. Thereafter, the next is conformational change in GP120 promotes binding to chemokines receptor. Then GP41 fusion peptides happens. Then a conformation change of GP120 exposes fusion peptide, which insert into T cell membrane. Then the fusion of viral and cell membrane happen. So transcription of the gene of integrated DNA provirus is regulated by LTR upstream of the virus structure, genes and cytokine and other stimuli that activate T cells and macrophage, enhance viral gene transcription. The LTRs contain polyadenylation signal sequence of the Tata box promoter sequence and the binding site for the 2 host cell transcription factor. Nuclear factor kappa beta and SP1 initiation of HIV genes transcription in T cell is linked to activation of the T cells by antigen or cytokines. For example, a polyclonal activator of T cells such as photohemagglutinin and cytokines such as interleukin-2 tumor necrosis factor and the lymphotoxin stimulate HIV gene expression in infected T cell. Various macrophage activated cytokines, including interferons gamma and the granulocyte macrophage colony stimulating factor, stimulate HIV genes expression and viral replication is infected monocyte and macrophage. T cell receptor and cytokine stimulation of HIV gene transcription probably involve the activation of nuclear factor kappa beta and its binding to sequence in viral LTR. This phenomena is significant in pathogenesis of AIDS because normal response of a latent infected T cells to a microbes may be way in which HIV latence is ended and virus production begins. The TAT protein is required for HIV gene expression and active by enhancing the production of complete viral messenger RNA transcript. Even the presence of optimal signal to initiate transcription, few, if any, HIV messenger RNA molecules are actually synthesized without the action of the TAT because transcription of HIV genes by mammalian RNA polymerase is inefficient, and the polymerase complex usually stops before messenger RNA is completed. TAT allow DNA-dependent RNA polymerase to retain bond to viral DNA molecule long enough for transcription to be completed, and thus produce a functional viral RNA. Synthesis of mature infectious viral particle begin after full length with viral RNA transcript are produced, and the viral genes are expressed as proteins. The messenger RNAs encoding the virus gene HIV proteins are derived from a single full genome length transcript by differential splicing events. HIV genes expression may be divided into an L stage, during which regulatory genes are expressed, and late stage, during which structural genes are expressed and full-length viral genomes are packed. The Rev, TAT, and NEF proteins are L gene products encoded by full splicing messenger RNA that are exported from the nucleus and translated into protein in cytoplasm soon after infection of the cells. Late genes include MV, GAG, and poly, which encode the structural component of viruses and are translated from the simply splicing or unsplicing RNA. The Rev proteins initiate the switch from early to late gene expression by promoting the export of these incompletely splicing late genes RNA out of the nucleus. The Pol genes product is a precursor protein that is sequential cleaved to form a reverse transcriptase, protease, ribonuclease, and integrase enzymes. As mentioned earlier, reverse transcriptase and integrase proteins are required to produce a DNA copy of viral RNA genome and to integrate it as provirus into the host genomes. The GAG gene encodes a 55 kilodalton protein that is proteotically cleaved into P24 capsid protein, P17 matrix proteins, P7 nucleocapsid proteins, P6 domain, and 2 spacer peptide SP1, SP2 by the action of viral protease encoded by Pol gene. These GAG-derived peptides proteins are required for the assembly of infection viral particles. The primary product of ENV genes is a 160 kilodalton glycoproteins that is cleaved by cellular protease within endoplasmic reticulum into the GAG120 and GAG140 protein required for HIV binding to cells. As discussed earlier, current antiviral therapy for HIV disease include inhibitor of enzymes, reverse transcriptase, protease, and integrase. After the transcription, viral genes, viral proteins are synthesized in cytoplasm. Assembly of infectious viral particle then begin by packing full-length RNA transcript of the proviral genome within nucleoproteins complex that includes a GAG core proteins and Pol-encoded enzymes required for next cycle of integration. This nucleoprotein complex then buds from the plasma membrane, capturing INV and host glycoprotein as part of its envelope. The rate of virus production can reach sufficiently high level to cause cell death, as discussed later. Pathogenesis of HIV infection and AIDS. HIV disease begins with acute infection, which is only partly controlled by the host immune response, and advances to chronic progressive infection of peripheral lymphoid tissue. The virus typically enters through mucosal epithelial. The subsequent event is the infection. In the infection can be divided into several phases. Acute early infection is characterized by infection of activated CD4 T cells in mucosal lymphoid tissue and death of many infected CD4 T cells. A larger number of activated and memory CD4 T cells reside at mucosal sites, primarily in the gastrointestinal tract, and can be infected by HIV. Within 2 to 4 weeks of infection, a large fraction of mucosal CD4 T cells may be destroyed. The transition from the acute phase to chronic phase of infection is accompanied by dissemination of the viruses, viremia, and development of the host adaptive immune response. Dendritic cell is a dendritic cell in epithelial at site of virus entry, capture the virus and then migrate to the lymph nodes. The dendritic cell expresses a protein with mannose binding lectin domain called the DC signs, which may be particularly important in binding the HIV envelope and transporting the viruses and mediating trans-infection of CD4 T cells. Once in lymph nodes tissue, the dendritic cell may pass HIV to the CD4 T cell through direct cell contact. Within days after the first exposure to HIV, viral replication can be detected in lymph nodes and gut lymphoid tissue. This replication leads to viremia, during which a large number of HIV particles are present in patients' bloods, accompanied by acute HIV syndrome that include variety of non-specific signs and symptoms, typically of many viral infections, described later. The viremia allows the virus to to disseminate throughout the body and to infect helper T cells, macrophage and dendritic cell in peripheral lymphoid tissue. As HIV infection spread to adaptive immune system, mount both as HIV infection spread, the adaptive immune system mount both humoral and cell-mediated immune response directed at viral antigens, which we will describe later. The immune response particularly control the infection and viral production, as such control is reflected by a drop in viremia to low detectable level of by approximately 12 weeks after the primary exposure. In the next chronic phase of the disease, lymph nodes and spleen and gastrointestinal tract are site of continuous HIV replication and cell destruction. During these periods of disease, the immune system remains competent at handling most infection with opportunistic microbes, and few or no clinical manifestation of HIV infection are present. Therefore, this phase of HIV phase is called the clinical latency period. Although the majority of peripheral blood T cells do not harbor the virus, destruction of CD4 T cells within lymphoid tissue steadily progresses during the latent period, and the number of circulating blood CD4 T cells steadily decline. More than 90% of the body, approximately 10 times 12 T cells, are normally found in peripheral and mucosal lymphoid tissue, and it is estimated that HIV destroys up to 1 to 2 times 10 power 9 CD4 T cell every day. Early in the course of the disease, the individual may continue to make new CD4 T cells, and therefore these cells can be replaced almost as quickly as they are destroyed. At this stage, up to 10% of CD4 T cell in lymphoid organs may be infected, but the number of circulating CD4 T cells that are infected at any one time may be less than 0.1 of the total CD4 T cell in individual. Eventually, over a period of years, the continuous cycle of viral infection, T cell death, new infection lead to the inexorable loss of CD4 T cells from lymphoid tissue and circulation. Mechanism of immunodeficiency caused by HIV. HIV infection ultimately results in impaired function of both adaptive and innate immune system. The most prominent defects are in cell-mediated immunity, which result mainly from a destruction of CD4 T cells. Both infected and possibly non-infected CD4 T cells may be lost. A major cause of the loss of CD4 T cell in HIV-infected individual is direct effect of infection for these cells by HIV. Death of the CD4 T cells is associated with production of virus in infected cell and may contribute to the decline in number of these cells. Several direct toxic effects of HIV on infected CD4 T cell have been described. The process of virus production with expression of GP41 in the plasma membrane and binding of the viral particle may lead to the increased plasma membrane permeability and influx of the lethal amount of calcium, which induce apoptosis and osmotic lysis of the cells caused by influx of the water. Viral production can interfere with cellular protein synthesis and thereby lead to the cell death. Mechanism in addition to viral-induced deaths of infected CD4 T cells have been proposed for depletion and functional impairment of this cell in HIV-infected individual. One mechanism is related to chronic activation of an infected cell by the infections that are common in patients infected with HIV. Also by cytokine produced in response to this infection. Chronic activation of T cells may predispose them to apoptosis. The molecular pathway involved in these types of activation induced cell death is not yet defined specifically in HIV context. In general, many activated CD4 T cells, especially T helper 1 cells, may be eliminated by FACS ligand fast interaction. Apoptotic death of activated lymphocyte may occur for the observation that the loss of T cells greatly exceed the number of HIV-infected cells. As mentioned earlier, HIV-specific cytotoxic T lymphocytes are present in many patients with AIDS, and those cells may kill infected CD4 T cells, and some abortively infected cells might die by pyroptosis. In addition, antibody against HIV envelope proteins may bind to HIV-infected CD4 T cells and target the cell for ADC. Binding of the GR120 to newly synthesized intracellular CD4 may interfere with the normal protein process in the endoplasmic reticulum and block cell surface expression of CD4, making the cell incapable of responding to antigenic stimulation. The relative importance of this indirect mechanism of CD4 T cell depletion in HIV-infected patients is uncertain and controversial. So we have the diagram again, the progression of HIV infection. The progression of HIV infection correlates with the spread of viruses from the site of infection to lymphoid tissue throughout the body. Immune response of the host temporarily controls the acute infection, but does not prevent establishment of chronic infection of the cell and lymphoid tissue. Cytokine stimuli induced by the microbe serve to enhance HIV production and progression of AIDS. So we have the diagram, the infection in the mucosa. They have been told that they're mostly common in the mucosa. Then the dendritic cell take the cell to CD4 T cells, memory CD4 T cells, they are dying. The infection established in lymphoid tissue, lymphoid nodes, a spread of infection throughout the body, viremia increases, more antibodies, cells are killed, and more infection is happening, to the point whereby there is destruction of lymphoid tissue, depletion of CD4 T cells will happen. Functional defect in immune system of HIV-infected individual exacerbate the immune deficiency caused by depletion of CD4 T cells. These functional defects include a decrease in T cell response to antigens and non-neutralizing and less vigorous humoral immunity response. Even though total serum IG level may be elevated, the defect may be as a result of direct effect of HIV infection on CD4 T cells, including the effect of a soluble GPR120 released from infected cell binding to an infected cell. For example, CD4 that has bound to GP120 may not be available to interact with class 2 MSC molecules on antigen-presenting cell, and thus T cell response to antigen will be inhibited. Also, the GP120 binding to CD4 may deliver a signal that downregulate helper T cell function. Some studies have demonstrated that patients with HIV infection have increased the number of CD4, CD25 regulatory T cell, but it is not yet clear if this is consistent finding or if these cells actually contribute to the defective immunity. Macrophage, dendritic cell, and follicular dendritic cells may be infected or injured by HIV, and resulting in abnormality of these cells also contribute to the progression of immunodeficiency. Although macrophages are susceptible to HIV infection, they are relatively resistant to cytopathic effect of HIV. Macrophages also have been infected by GP120, GP41 independent routes such as phagocytosis of viruses or other infected cell, Fc receptor-mediated endocytosis of antibody-coated HIV virions. Because macrophages can be infected but are not generally killed by HIV, they may become reservoir for virus. In fact, the quantity of macrophages associated with HIV exceed T-cell associated virus in some virus from the patient with AIDS. Notably, the brain, HIV-infected macrophage may be impaired in antigen presentation, function, and cytokine secretion. So we have a clinical cause of HIV disease, a plasma viremia, blood CD4 T cell count, and clinical stages of disease. Approximately 12 weeks after infection, the blood-borne virus plasma viremia is reduced to very low level, detectable only by sensitive reverse transcriptase polymerase chain reactions, and stay this way for many years. Nevertheless, CD4 T cells count steadily decline during the clinical latence period because of active viral replication and T cell infection in lymph nodes. When CD4 T cells counts drop below the critical level of 200 per milliliter, the risk of infection and other clinical features of AIDS is high. Immune response to HIV infection, a cytotoxic T lymphocytes response to HIV is detectable by 2 to 3 weeks after the initial infection and peaks by 9 to 12 weeks. Marked expansion of viral-specific CD8 T cells occur during this time, and up to 10% of the patient's cytotoxic T lymphocyte may be HIV-specific at 12 weeks. The humoral immunity response to HIV peaks at about 12 weeks. Let us return a little bit. So if you're following, these are the things that will be happening. In terms of CD4, they will actually gradually count down. So we have primary infection here, so they will be as high. Then your body is mounting immune. So why the dissemination of virus seedling lymphoids, you see there is increase, but the primary infection will be going down because of immunity. So B is about immunity, I think. So antiretroviral antibody, specific. So you see the immunity are going up, and the antibody and the infection will be going down. The dendritic cell can also capture HIV and can possibly also be infected, like macrophage. Dendritic cells are not directly injured by HIV infection. However, these cells form intimate contact with naive T cells during a course of antigen presentation. It is proposed that dendritic cells infect naive T cells during this encounter, and this may be a pathway for spread of infection. Follicular dendritic cell in germinal center of lymph nodes and spleen trap large amount of HIV on their surface in part of Fc receptor-mediated binding of antibody-coated viruses. Although follicular dendritic cells are not efficiently infected, they contribute to the pathogenesis of HIV-associated immunodeficiencies in at least 2 ways. First, the follicular dendritic cell surface is reservoir for HIV that can infect macrophage and CD4 T cells in lymph nodes. Second, the normal function of follicular dendritic cell in immune response are impaired, and they may eventually be destroyed by virus. Although the mechanism of HIV-induced death for the follicular dendricyte are not understood, the net result of loss of follicular dendricyte network in the lymph nodes and spleen is profound dissolution of architecture of peripheral lymph node system. T follicular helper cells in the germinal center may also contribute to HIV reservoir discussed in the following section. HIV reservoir and viral turnover. The virus detected in patient blood is produced mostly by short-lived infected CD4 T cells and a small amount of other infected cells. 3 phases of decay of plasma viremia have been observed in patients treated with the antiretroviral drugs and predicted by mathematical modeling. And these decay curves have been used to summarize the distribution of HIV in different cellular reservoirs. More than 90% of the plasma virus is thought to be produced by activated and memory CD4 T cells that are a major reservoir and the source of virus in infected patients. About 2/3 of the HIV reservoir exists in the mucosal system, mainly in gastrointestinal tract, and about 1/3 is in lymph nodes. Although there are ongoing attempts to enhance host immunity and clear the virus completely from infected individuals, often referred to the HIV cure, the resilience of HIV reservoir have proved a confounding factor. Many latent viruses within the reservoir remain relatively protected from the host immune system. Latent viruses in T follicular cells in the light of zones of germinal have been assumed to be protected from cytotoxic CD4 T cells, CD8 T cells. Some plasma virus may be produced by macrophages, which have a slower turnover, half-life of approximately 2 weeks. It is hypothesized that very small fraction of viruses, perhaps as little as 1%, is present in latent infected memory T cells. Because of the long lifespan of memory cells, it could take decades for this reservoir of virus to be eliminated, even if a new round of infection were blocked. Clinical features of HIV disease. A vast amount of information have accumulated about epidemiology and clinical cause of HIV infection. The antiretroviral drugs therapy is improving. Many of the clinical manifestations are changing. In the following section, we will describe the clinical features of HIV infection and refer the changing picture when relevant. Transmission of HIV and epidemiology of AIDS. HIV is transmitted from one individual to another by 3 major routes. 1, sexual contact is the most frequent mode of transmission, either between heterosexual couples, the most frequent mode of transmission in Africa and Asia, or between men having sex with men in sub-Saharan Africa, where the infection rate is highest in the world, estimated to be several thousand new cases every day. More than half of infected individuals are women. Mother-to-child transmission of HIV account for the majority of the pediatric case of AIDS. This type of transmission occurs most frequently in utero or during childbirth, although transmission through breast milk is also possible. Inoculation of recipient with infected blood or blood product is also a frequent mode of HIV transmission. Needle shared by intravenous drugs user account for the most causes of this form of transmission. HIV can remain infectious in used infected needle for 6 weeks in temperature climate. With the advent of routine laboratory screening, transfusion of blood or blood products in a clinical setting account for negligible portions of HIV infections. Clinical cause of HIV infection. The cause of HIV disease can be followed by measuring amount of viruses in patient plasma and by blood CD4 cell counts. 3 phases of disease are recognized. The acute phase, also called acute HIV syndrome, is the period of viremia characterized by non-specific symptom of infection. It develops in 50% to 7% of infected adults, typically 3 to 6 weeks after infection. There is a spike of plasma virus and a modest reduction in CD4 T cell count, but the number of blood CD4 T cells are often returned to normal. In many patients, however, the infection is occult and there is no symptoms. The chronic phase of clinical alertness may last for many years. During this time, the virus is contained within lymphoid tissue, and loss of CD4 T cell is corrected by replacement from the progenitor. Patient are symptomatic or suffer from minor infection. Within 2 to 6 months after infection, the concentration of plasma virus stabilizes at a particular set point, which differ among the patients. The level of the viral set point and number of the blood CD4 T cells are clinically useful predictor of progressions of the disease. As the disease progression, patients become susceptible to other infection, and immune response to these infections may stimulate HIV production and accelerate the destruction of lymphoid tissue. As discussed earlier, HIV genome transcription can be enhanced by stimuli that activate infected T cells, such as antigen and virus cytokines. Cytokines such as tumor necrosis factor, which are produced during the innate immune response to microbial infection, are particularly effective in boosting HIV production. Thus, as immune system attempt to eradicate other microbes, it brings about its own destruction by HIV. A tragic example that has been called subversion from within. HIV disease progressed to a final phase called AIDS when the blood CD4 T cell count dropped between 200 cells millimeter cube. HIV viremia may climb dramatically as viral replication accelerates unchecked in reservoir other than T cells. Patients with AIDS suffer from combination of opportunistic infection, neoplasia, cachexia, HIV wasting syndrome, kidney failure, HIV nephropathy, and CNS disease, AIDS encephalopathy, now called HIV-associated neurocognitive disorder, or HAND. Because CD4 T cell helper are essential for both cell-mediated and humoral immune response to virus microbes, the loss of these lymphocytes is the main reason for the patient with AIDS to become susceptible to many different types of infection. Furthermore, many of the tumors that arise in patients with AIDS have viral cause, and their prevalence in setting of AIDS reflect the inability of HIV-infected patients to mount an effective immune response against oncogene viruses. Cachexia is often seen in patients with chronic inflammatory disease and may result from effect of inflammatory cytokines such as tumor necrosis factor on appetite and metabolisms. The CNS disease in AIDS may be due to neurological damage of virus by shed of viral proteins, such as G120 TAT, as well as effects of cytokine elaborated by infected microbial cells. Clinical feature of HIV infections. Phase disease clinical feature. Acute HIV disease, it involves fever, headache, sore throats, pharyngitis, generalized lymphadenopathy, and rashes. Clinical latence, here there is decline of CD4 T cell counts. Then in AIDS, opportunistic infection, including protozoa infection, for example, Toxoplasma, Cryptosporidium, bacterial infection, for example, Mycobacterium avium, Nocodia, Salmonella, fungal infection like Candida, Cryptococcus neoformis, Candidiasis immitis, Histoplasma capsulatum, and pneumocystis, virus like cytomegalovirus, herpes simplex, varicella zoster, tumor like lymphoma, including Epstein-Barr associated B cell lymphoma, Kaposi sarcoma, cervical carcinoma, neurocognitive disorder, wasting syndrome. All these one will happen in AIDS. Although this summary of the clinical causes is true for the most severe cases, the rate of progression of disease is highly variable, and some individuals are long-term non-progressor. The possible mechanism underlying the long-term non-progressor are described later. Importantly, antiviral therapy, which is now increasingly available, has changed the course of disease and greatly reduced the incidence of severe opportunistic infection, such as pneumocystis, and tumors such as Kaposi sarcoma. Immune response to HIV. Innate immune to HIV and host restriction factor. Host restriction factor inhibit viral infection, and many viral proteins have evolved to counter this restriction factor. Host restriction factor are best appreciated in the overall context of innate immune response to HIV. HIV is sensed by a number of pattern recognition receptors, including toll-like receptor, RIG1, two key sensors that recognize viral reverse transcriptional product early in infection are IF116, interferon-inducible protein 16, and C-gas, cyclic GMP-AMP synthetase. Both discussed in chapter 4. IF, which is interferons-inducible proteins, which is interferon-inducible proteins 16, can bind to HIV-derived copy of DNA and signal via a stimulator of interferon genes adapter, the TKB proteins kinase, and IRF3 and IRF7 transcription factor. This signal induces type 1 interferon production and the expression of the host restriction factor such as ApoBec, TRIM5 alpha, SAM-HD. The terethrin described next. Terethrin is the host factor that prevents virion release in certain cell types. It prevents the pitching off of the certain viruses, including HIV, and its inhibition of the budding process can be antagonized by HIV protein called VPU. Host cell incorporate certain restriction factors, messenger RNA, certain restriction factor into the virus particle, including ApoBec, apolipoprotein B messenger RNA editing enzymes, catalytic peptide like 3 proteins. This protein is cytidines, deaminase that interfere with viral replication in infected cell. The HIV VIF proteins help to target ApoBec proteins by ubiquitinations and proteasomal degradation and thus promote viral replication. In infected cell, another important host restriction factor is TRIM5 alpha, a member of TRIM, tripartite motif family of ubiquitin E3 ligase. TRIM interact with HIV capsid proteins to cause premature uncoating and viruses of the viruses and proteasomal degradation of viral reverse transcriptase complex. It can also block nuclear translocation of viral pre-integration complexes. SAMD or SAM domain and HD domain 1 is the host enzyme that hydrolyze and deplete intracellular deoxynucleotide triphosphate and thus prevent synthesis of viral DNA by reverse transcriptions. HIV-2 virus strain produce a protein called VPX that antagonizes the depleting activity of SAM-HD1. Many other innate immune responses against HIV have been described. These include production of the production of antimicrobial peptides, defensin and activation of natural killer cells, dendritic cell, particularly plasmacytocytes, and dendritic cell producing interferon gamma and the complement system. Interferons, not interferon gamma. Adaptive immune response to HIV. HIV-specific humoral and cell-mediated immune response develop after infection but generally provide limited protection. The L immune response to HIV infection is in fact similar to response to other viruses and serves to clear most of the virus present in the blood and in circulating T cell. Nevertheless, it is clear that these immune response fail to eradicate HIV, and the infection eventually overwhelm the immune system in most untreated individuals. Despite the poor effectiveness of viral immune response, it is important to characterize them for three reasons. First, immune response may be detrimental to host. For example, by stimulating uptake of opsonizing virus into an infected cell by Fc-mediated endocytosis. Second, antibody against HIV are diagnostic marker for HIV infection that are widely used for screening purpose. Third, the design of effective vaccine for immunization against HIV require knowledge and type of immune response that are most likely to be protective. Related of protection. The initial adaptive immune response to HIV infection is characterized by expression of CD8 T cells specific for HIV peptide. As many as 10% of them or more of circulating CD8 T cells may be specific for HIV, GAG, and other viral proteins during acute infection. These CTL, cytotoxic T lymphocyte, control infection in L phase, but ultimately prove ineffective because of emergence of viral escape mutants variant with the mutated antigens. CD4 T cells also respond to the virus, and these CD4 T cells may contribute to viral control in number of ways. An effective CD4 T cell response is required as source of helper for generation of CD T cells, but CD4 T cells have also been shown to kill HIV-infected cell. The importance of CTL as cytotoxic T lymphocyte response in HIV control is underscored by evolution of virus under immune pressure, resulting in viral isolate that have lost their original as CTL epitope. The evolution of viruses are also in loss of epitope recognized by CD4 T cell, indicating that both CD4 and CD8 and CD4 T cells contribute to the host defense against the virus. Antibody response to a variety of HIV antigen are detectable within 6 to 9 weeks after infection. Most immunological HIV molecules that elicit the antibody response appear to be the envelope glycoproteins, and high titer of anti-G120 and anti-G41 antibody are present in most HIV-infected individuals. Other antibodies found frequently in patients' sera include antibodies to P24 reverse transcriptase, GAG, and POL products. The effects of these antibodies on the clinical cause of HIV infection is uncertain. The L antibodies are generally not neutralizing and are thus poor inhibitor of viral effectivity or cytopathic effect. Neutralizing antibodies against G120 develop 2 to 3 months after primary infection, but even these antibodies cannot cope with virus that is able to rapidly change the most immunodominant epitope of its envelope glycoprotein. Sequencing of antibody heavy and light chain genes from G140 specific B cell in subjects who have been infected with HIV-1 for a few years have revealed the presence of broadly neutralizing antibodies. Intriguingly, for unknown reason, only approximately 10% to 15% of chronically infected individuals develop broadly neutralizing antibody. These antibodies bind to the site of viral proteins that the virus cannot afford to mutate, such as CD4 binding site of GP140. They are therefore effective in clearing the virus. A striking feature of all of these antibodies is that they have been selected after extensive somatic hypermutations, implying helper T cells depending antibody response. The implication that the starting naive HIV specific B cell repertoire primarily consists of B cells whose antigen receptor bind weakly to certain antigens epitopes such as CD4 binding site of G140. Many rounds of somatic hypermutation and selection that may occur in long-stand infection can eventually generate B cell population that bind with high affinity in the original weakly recognized epitope. One of the goals of vaccination is to generate such high affinity broadly neutralizing antibody, but inducing the high rate of somatic hypermutation necessary to achieve this has proved a challenge. This has not been achieved with any consistency. Mechanism of immune evasion by HIV. Mechanism of immune evasion by HIV. HIV is prototype of infection pathogen that evade host defense by destroying the immune system. We have earlier considered some mechanism by which this virus evade host restriction factors and innate immunity. In addition, several features of HIV may help the virus to evade adaptive immunity. HIV has extremely high mutation rate because of error-prone reverse transcriptions, and in this way, it may evade detection of antibodies or T cells generated in response to viral proteins. A region of the G120 molecules called V3 groups is one of the most antigenically variable part of the virus. It differs even in HIV isolate taken from the same individual at different time. Many epitopes of the virus that could potentially serve as target of broadly neutralizing antibody are shielded by bulk N-linked sugar that make up what is known as HIV glycan shield. HIV-infected cells may evade cytotoxic T lymphocyte through downregulation of class 1 MHC molecule expression. The HIV NEF proteins inhibit expression of class 1 molecules mainly by promoting internalization of these molecules. Other mechanisms of inhibiting cell-mediated immunes have been demonstrated in some cases. I mentioned earlier, these include preferential inhibition of T helper 1 cytokine, activation of regulatory T cells, suppression of dendritic cells function. Mechanism of this action of virus and their pathogenic significance are not established. Elite controller and long-term non-progressor, a possible role of host genes. Although most individuals infected with HIV eventually develop AIDS, approximately 1% of individuals who are infected do not develop clinical disease. Such individuals have high CD4 and CD8 T cell count, do not require therapy, and may have persistent viremia but no disease far as at least 10 to 15 years. On the basis of the degree of viremia, this group can be divided in two subsets. Long-term non-progressor have detectable viremia of approximately 5,000 copies of HIV-1 RNA per millimeter of the blood, and much smaller subset of elite controller maintain viral loads of approximately 50 copies of or less of HIV RNA per millimeter of the blood. There is considerable interest in the understanding the genetic basis of HIV control by examining these cohort of individuals in detail. So far, a strong role of MHC locus in protecting individual and preventing progressor has been suggested by genetic association studies. Specific HLA class 1 lock has been linked to the absence of disease progression. Some elite controller generate strong CD8 T cell response to highly conserved peptide in virus so that the virus cannot mutate without losing effectivity. We have previously mentioned the importance of the inheritance of CCR5 homozygous 32PB deletion in protection from the infection. Other genetic factors contribute to this resistance are likely to be revealed in coming years. Treatment and prevention of AIDS and vaccine development. Active research efforts have been aimed at developing a drug that interferes with the viral life cycle. Treatment of HIV infection and AIDS now typically involves the administration of 3 antiviral drugs used in combination that target viral molecule for which no human homologue exists. The first antiviral drugs to be widely used were nucleoside analogs that bind to viral reverse transcriptase and inhibit its activity. These drugs include deoxythymidine nucleoside analogs such as AZT, 3-azidodeoxythymidine, deoxycytidine nucleoside analogs, deoxyadenosine analogs. When these drugs are used alone, they are often effective in significantly reduce the plasma HIV RNA level for several months to years, but they usually do not halt the progression of HIV-induced disease, largely because of evolution of virus with mutated form of reverse transcriptors that are resistant to drug. Nucleoside reverse transcriptase inhibitor, non-nucleoside reverse transcriptase inhibitor directly bind to the enzymes and inhibit its fusion. Viral protease inhibitors have been developed to block processing of the precursor protein into the mature viral capsid and core protein. When these protease inhibitors are used alone, mutant virus resistant to their effector emerge. However, protease inhibitors are now component of 3 drug therapeutic regimes with 2 different reverse transcriptase inhibitors. These triple drugs, commonly referred to as highly active antiretroviral therapy, HAT, or antiretroviral therapy IRT, have proved to be effective in reducing plasma viral RNA to undetectable level in most treated patients for years. An integrase inhibitor is now also available for antiviral therapy. Entry inhibitor, which prevent viral entry to targeted cell CD4 and CCR5 on the host cell, are another novel category of therapeutic. Fusion inhibitor are the drug that target GP41 and prevent fusion of the viral envelope with the host cell plasma membrane. Although antiviral therapy has reduced the viral titer to below detectable for up to 10 years in some patients, it's unlikely that such a treatment can eliminate the viral from the reservoir, especially long-lived infected cells. The resistance to drugs may ultimately develop. Other formidable problems are associated with these new drugs therapy, which will impair their effective use in many parts of the world, including high expenses and significant adverse effects. Furthermore, in some patients, the virus evolved to become resistant to the drug being used. This problem is often managed by sequencing the viral genomes to identify mutations that may make the virus drug resistant and change the drug regime accordingly. Inconsistent use of these drugs by patients is also the major problems. Pre-exposure prophylaxis PrEP using a single pill that contains 2 mechanistically different reverse transcriptase inhibitor is designed to protect individuals who do not have HIV but who have high risk of getting the disease. Although PrEP have been effective in some contexts, its inconsistent use has proved a major problem, and the rollout of PrEP has so far not blueprint for epidemic in those parts of the world where the need is greatest. A proportion of patients receiving ART experience aberrant manifestation of immune reconstitution, namely overexperienced inflammation that may be triggered by recognition by immune system of existing pathogens. It is usually accompanied by restoration of CD4 T cells counts and decline in viral loads. This clinical phenomena is called immunoreconstitution inflammatory syndrome, IRIS. The individual infection experienced by patients with AIDS are treated with the appropriate prophylactic antibiotics and supportive measures. More aggressive antibiotic therapy is often required than for similar infection in less compromised hosts. Although antiviral therapy had led to the longer survival rates after HIV infection, chronic HIV infection is associated with the increased risk of non-AIDS related morbidity and mortality than seen in the age-matched uninfected individual. In these patients, there is a well-documented increase in cardiovascular disease, liver disorder, disorder of neurocognitions, kidney disease, and some cancers. Chronic HIV is now recognized as a chronic inflammatory disease with the manifestation that may be linked to local or systemic inflammation. This syndrome is the focus of many ongoing studies. Efforts at prevention of HIV infection are extremely important and potentially effective in controlling the HIV epidemic. In the United States, the routine screening of blood products for evidence of donor HIV infection has already reduced the risk of the mode of transmission to negligible level. Various public health measures to increase condom use and reduce the use of contaminated needle, intravenous drug use are now widespread. Perhaps the most effective effects at prevention and are campaigning to increase public awareness of HIV. Clinical trials are showing that administration of antiretroviral drugs to pregnant mother is effective at preventing infection of the newborns. Prophylactic use of these drugs in high-risk patients also reduce the rate of infections, as mentioned. The development of an effective vaccine against HIV is a priority for biomedical research institutions worldwide. The task has been complicated by ability of virus to mutate and vary many of its immunogenic antigens. It is likely that an effective vaccine will have to stimulate both humoral and cell-mediated response to viral antigens. To achieve this goal, several approaches are being tried for HIV vaccine development. Much of the primary work have involved infection of monkey with simian immunodeficiency virus, and effective vaccine against HIV, simian immunovirus have been developed. This success is encouraging because simian immunovirus is molecularly closely related to HIV, and cause of disease in monkey that is similar to AIDS is in human. Various live viruses vaccines have been tested in hope that they will induce strong cytotoxic T lymphocyte response. Such vaccines include non-virulent recombinant hybrid virus, non-hybrid virus composed of the pasty SIV and part of HIV sequence, or virus that has been attenuated by deletion in one of the more part of viral genome, such as the NEF genes. One concern with live virus vaccine is their potential to cause disease if they are not completely attenuated, and possibility to recombine with wild type HIV to produce a pathogenic variant. Another approach that avoid this safety concern but retain efficacy in inducing cytotoxic T lymphocyte mediated immunity in the use of live recombinant and non-HIV viral carrying HIV genes. Many DNA vaccines also have been studied. These vaccines are composed of recombination of structural and regulatory genes of CIV or HIV packed in mammalian DNA expression vector. T-cell vaccine focused on conserved regions of the viral proteins represent an interesting approach that is being undertaken and is currently in its early stages. Most current vaccination attempts involve recombinant subunit vaccine with HIV trimers and are aimed at generating broadly neutralizing antibody to GP140. A broadly neutralizing antibody is one that can prevent the infectivity of a large number of HIV strains, and such as antibody would therefore have to recognize some part of GP140 as such as CD4 binding site, for instance, which is highly conserved. Human trials are currently underway with recombinant GP140 trimer in adenoviral vector based on the adenovirus strain with low human seroprevalence. An interesting and inventive approach has involved the identification of rearranged IG genes that encode broadly neutralizing monoclonal human antibody derived from individual clonal B cells isolated from a subsequent of chronic progressors. These antibodies arise too late to be useful for patients who generate them, but they provide lessons about how such antibodies evolved in vivo and have proved to be important for current vaccine generation attempt. It is possible to identify naive B cells that have rearranged Ig heavy and light chains genes that somatically mutate after activation and give rise to these specific protection broadly neutralizing antibody. These advances have led to the rational design of GP140 immunogens based on structural biology that can be potentially activate B cells in a way that facilitates stepwise evolution of somatically mutated broadly neutralizing antibody. These promising approach have surprisingly proved successful in mice engineered to express human Ig genes and may enter the clinical realm one day. Other approaches for protection against HIV include the use of passive immunity with mixture of broadly neutralizing antibodies derived as described earlier that have been recombinantly generated for clinical use. Early clinical studies have been promising. Vectored immunoprophylactic is a form of immunity in which a protein that can mediate immune protection is synthesized in the host typically after injection of specific DNA into skeletal muscle. In one approach that is currently being attempted in clinical trial, the Ig heavy and light chain genes that encode a broadly neutralizing antibody against HIV have been cloned into an adenovirus-associated virus vector. This DNA have been injected into the muscle of the volunteer. An alternative approach that has worked well is simian model. In simian model is expression in the host of fusion genes made up of CD4 Ig linked to small CCR5 mimetic sulfopeptide. This fusion protein effectively prevent the entry of CIMV into the CD4 T cell. It is possible that if vaccine against HIV does not prove successful, vectored immunoprophylactic approach may be used to staunch the spread of HIV. Summary. Immunodeficiency disease are caused by congenital or acquired defect in lymphocytes, phagocytes, and other mediator of adaptive and innate immunity. There are several diseases. These diseases are associated with increased susceptibility to infection and nature of severity of which depend largely on which component of immune system is abnormal and extent of abnormalities. Disorder of innate immune include defective microbial killing by phagocyte, chronic granulomatous diseases, Cheddar-Higashi syndrome, leukocyte migrations and adhesions, leukocyte adhesion deficiency, toll-like receptor signaling and complements. Severe combined immunodeficiency include defect in lymphocyte development that affect both B and T cells and are used by defective cytokine signaling, abnormal purine metabolic defects, VDJ recombinants, and mutation that affect T cell maturation. Here I meant are caused. Let me repeat. Severe combined immunodeficiency include defect in lymphocyte development that affect both T and B cells and are caused by defective cytokine signaling, abnormal purine metabolism, defect in VDJ recombination, and mutation that affect T cell maturation. Antibody immunodeficiency include disease caused by defective B cell maturation or activation and the defect in T and B cell decollaboration, X-linked hyper IgM syndrome. T cell immunodeficiency include disease in which the expression of the major histocompatibility complex molecule is defective, T cell signaling disorder, and rare disease involving cytotoxic T lymphocyte and natural killer cell function. Treatment of congenital immunodeficiency involve transfusion of antibodies, stem cell transplantations, and enzyme replacement. Gene therapy may offer improved treatment in future. Acquired immunodeficiencies are caused by infection, malnutrition, disseminated cancer, and immunosuppressive therapy for transplant rejection or autoimmune disease. AIDS is a severe immunodeficiency caused by infection with HIV. This retrovirus infects CD4 T lymphocyte, macrophage, and dendritic cells, and causes progressive dysfunction of the immune system. Most of immunodeficiency in AIDS can be attributable to the depletion of CD4 T cells. HIV enters cells by binding to both CD4 molecule and co-receptor of the chemokine receptor family. After it is inside the cell, the viral genome is reverse transcripted into DNA and incorporated into the cellular genome. Viral genes transcriptions and viral reproduction are stimulated by signal that normally activate the host cell. Production of virus is accompanied by death of infected cell. The acute phase of infection is characterized by death of activated and memory T cells in mucosal tissue and disseminated of the virus lymph node and of virus to lymph nodes. Dissemination of virus to lymph nodes. It is the subsequent latent virus. In the subsequent latent phase, there is low-level virus replication in lymphoid tissue and slow progression loss of T cells. Persistence activation of T cells promote their death, leading to rapid loss and immune deficiency in chronic phase of the infection. CD4 T cell depletion in HIV-infected individual is largely due to direct cytopathic effects of the virus. Several reservoirs of HIV exist in infected individuals, including short-lived activated CD4 T cells, long-lived macrophage follicular helper T cells, and very long-lived latent infected memory T cells, especially in mucocytes. HIV-induced depletion of CD4 T cells result in increased susceptibility to infection by a number of opportunistic microorganisms. In addition, HIV-infected patients have an increased incidence of tumor, particularly Kaposi sarcoma and Epstein-Barr virus-associated B-cell lymphoma and encephalopathy. The incidence of this complication has been greatly reduced by antiretroviral therapy. HIV has a high mutation rate, which allows the virus to evade the immune system response and become resistant to drug therapies. Genetic variability also poses the problem for design of an effective vaccine against HIV. HIV infection can be treated by a combination of inhibitor of viral enzymes. That marks the end of this discussion and also the end of this book reading. And if you're really interested, you can just start the book and read it again. It was just an adventure for me, like to try to challenge myself. It doesn't matter how much I have reserved in my head, but it was to challenge myself, read the whole book, and see different things that are available. And also, I know I might not have retained many information, but for sure, I know that there are a lot of things in immunology, and there are a lot of things that I need to know, I need to learn, and this has been a very nice experience for me. So I encourage you to buy the book and also read the book cover by cover. It will be an interesting journey of some hours that you have to spend. Yeah, all the best. This was Jeremiah Pusa, and this marked the end of this book discussion and reading. Thank you. In Dutch, they say, dankjewel. Haha.